Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today we're going to go over some of the non-enzymatic functions of proteins. Let's start with structural proteins. Structural proteins support and shape cells and tissues, contributing to their mechanical strength and integrity. The primary structural proteins in the body are collagen, elastin, keratin, actin, and tubulin. Structural proteins generally have a highly repetitive secondary structure and a supersecondary structure, which is a repetitive organization of secondary structural elements together, sometimes referred to as a motif. This regularity gives many structural proteins a fibrous nature. Let's start with collagen. Collagen is the most abundant protein in mammals. Collagen forms fibrils that provide tensile strength to connective tissues such as skin, tendons, and bones. It is found throughout the body and is important in providing strength and flexibility. It is a trihelical fiber, which means it has 3 left-handed helices woven together to form a secondary right-handed helix. Now elastin. Elastin is another important concept of the extracellular matrix of connective tissue. Its primary role is to stretch and then recoil, kind of like an elastic or an elastic band, and then it restores the original shape of the tissue. Now, keratins are intermediate filament proteins. They're found in epithelial cells. Keratins contribute to the mechanical integrity of the cell and also function as regulatory proteins. Keratin is found in hair, nails, and the epidermis. It provides protection and mechanical strength to epithelial tissues. Now let's talk about actin. Actin is a protein that makes up microfilaments and the thin filaments in myofibrils. It's the most abundant protein in eukaryotic cells. Actin proteins have a positive and a negative side, and this polarity allows motor proteins to travel unidirectionally along an actin filament, like a one-way street. Actin and tubulin together form the cytoskeleton with a dynamic network of filaments that are essential for cell shape, motility, and intracellular transport. That brings us to tubulin, which is a protein that makes up microtubules. Microtubules are important for providing structure, chromosome separation, mitosis and meiosis, and intracellular transport with kinesin and dynein. Like actin, tubulin also has polarity. The negative end of a microtubule is usually located adjacent to the nucleus, whereas the positive end is usually in the periphery of a cell. Now, some structural proteins also have motor functions in the presence of motor proteins. The motile cilia and flagella of bacteria and sperm are prime examples, as is the contraction of the sarcomere in muscle. Motor proteins also display enzymatic activity, acting as ATPases that power the conformational change necessary for motor function. Motor proteins have transient interactions with either actin or microtubules. Myosin is the primary motor protein that interacts with actin. In addition to its role as the thick filament in a myofibril, myosin can be involved in cellular transport. Each myosin subunit has a single head and neck. Movement at the neck is responsible for the power stroke of sarcomere contraction. Kinesin and dynein are the motor proteins associated with microtubules. They have 2 heads, at least one of which remains attached to tubulin at all times. Kinesins play key roles in aligning chromosomes during metaphase and depolymerizing microtubules during anaphase of mitosis. Dyneins are involved in the sliding movement of cilia and flagella. Both proteins are important for vesicle transport in the cell, but have opposite polarities. Kinesin brings vesicles towards the positive end of the microtubule, and dyneins bring vesicles towards the negative end. Now let's talk about binding proteins. While proteins exert enzymatic or structural functions within the cell, they also can have stabilizing functions in individual cells and in the body. Proteins that act in this way transport or sequester molecules by binding to them. Binding proteins include hemoglobin, calcium binding proteins, DNA binding proteins, and others. Each binding protein has an affinity curve for its molecule of interest. As we talked about in an earlier episode, the oxyhemoglobin dissociation curve is a well-known example of hemoglobin and oxygen. And each binding protein will have a different curve. As you may recall, the oxyhemoglobin dissociation curve is sigmoidal. This is because hemoglobin is cooperative. It has is 4 subunits. And in comparison, when you look at the myoglobin oxygen curve, you'll see it's hyperbolic. So this tells us that each binding protein has an affinity curve for its molecule of interest, and the curve differs depending on the goal of the binding protein. Now let's move on to cell adhesion molecules, or CAMs. These are proteins found on the surface of most cells and aid in binding the cell to the extracellular matrix or to other cells. While there are a number of different types of CAMs, they are all integral membrane proteins. Adhesion molecules can be classified into 3 major families: cadherins, integrins, and selectins. Cadherins are a group of glycoproteins that mediate calcium-dependent adhesion. They often hold similar cell types together, such as epithelial cells. Different cells usually have type-specific cadherins. For example, epithelial cells use E-cadherin, while nerve cells use N-cadherin. Also a quick tip, I remember cadherins as being calcium-dependent because the formula for calcium in the periodic table is Ca, and then if you just take the D, you get Cad, which is cadherin. So just think of that, and you'll think calcium-dependent. Moving on to integrins. Integrins are a group of proteins that will have 2 membrane-spanning chains called alpha and beta. These chains are very important in binding to and communicating with the extracellular matrix. Integrins play an important role in cellular signaling and can greatly impact cellular function by promoting cell division, apoptosis, or other processes. For example, integrin alpha 2b beta 3 allows platelets to stick to fibrinogen, which is a clotting factor, and this causes the activation of platelets to stabilize the clot. Other integrins are used for white blood cell migration, stabilization of epithelium on its basement membrane, and other processes. Selectins are unique because they bind to carbohydrate molecules that project from other cell surfaces. Of the 3 cell adhesion molecules we talked about today, selectins are the weakest bonds. They're expressed on white blood cells and the endothelial cells that line blood vessels. Like integrins, they play an important role in host defense, including inflammation and white blood cell migration. Now, let's talk about immunoglobulins. Immunoglobulins, also known as antibodies, are proteins produced by B cells that function to neutralize targets in the body, such as toxins and bacteria, and then recruit other cells to help eliminate the threat. Antibodies are Y-shaped proteins that are made up of 2 identical heavy chains and 2 identical light chains. The heavy and light chains are held together by disulfide linkages and non-covalent interactions. Now, each antibody has an antigen binding region at the tips of the Y. Within this region, there are specific polypeptide sequences that will bind one and only one specific antigenic sequence. The remaining part of the antibody molecule is known as the constant region, which is involved in recruitment and binding of other cells of the immune system, such as macrophages. So when antibodies bind to their targets, called antigens, they can cause one of 3 outcomes. The antigen can either be neutralized, which means the pathogen would be unable to exert its effect on the body. The pathogen could be marked so that other white blood cells could destroy it immediately, and this marking function is called opsonization. Or the antigen and antibody would go into large unsoluble protein complexes that can be phagocytized and digested by macrophages, and this is called agglutination. Essentially means they're just clumping together. Now, I know we've talked about a lot of these topics before, but just to have them in one episode I thought was a good idea. I also wanted to do the ion channels episode now, but I already talked about that earlier. So just make sure you watch the GPCR and the ion channels episode again after this. That's it for today. Next time we'll go over some practice questions.